lab at Henry Ford, correct? And we're live
Yes.
now. Okay. All right, everyone. Hello. It is Thursday, October 8th, and I am so honored to welcome Dr. Evelyn Jiagge, who has come to the United States since 2009, correct?
Yes.
From Ghana, and she's a specialist in triple negative breast cancer research. She has the Jiagge Lag at the Jiagge Lab at Henry Ford Health System. And we also want to thank Exact Sciences for sponsoring this time together. We have about 30 minutes to chat about Dr. Jiagge's work, and I'm going to just jump right in. So I wanted to know, Dr. Jiagge, what was it that prompted you to leave Ghana and come to the United States to further your studies and to begin this research project?
Thank you very much, Molly, and thank you for inviting me here. I'm really honored to be here to share my work with you. I'm a Ghanaian. I grew up in Ghana, schooled in Ghana. I'm a surgeon by training. And when I was taking care of breast cancer patients in Ghana, I guess what really resonated with me then was that the patients that we were seeing were not doing very well. Our patients were not surviving for as long as was being reported in the literature. We usually have patients, we see patients at very young age. I remember seeing a 17-year-old girl with advanced metastatic breast cancer. And we don't understand why our patients are so young, and we don't understand why they do as poorly as they do. But when you compare it to what is reported in the literature, breast cancer outcome is improving over the years, and women are doing better than they used to do previously. But that's not what we see. I remember seeing, I guess what the turning point for me was when I saw a 30-year-old woman who had come to my clinic. And she had a 3-year-old daughter, a 1-year-old son, and she was 4 months old pregnant. And she had a lump in her breast, and her breast was edematous, was swollen. She didn't know she had breast cancer. She thought she had an abscess. But when we did the biopsy, we realized it was advanced metastatic breast cancer in a woman who was pregnant and had 2 children who were less than 4 years old. And I remember when we told her that she had breast cancer, she fell down on the floor in the consulting room and started crying because she knew that that was a death sentence. And I wondered what we could do better for women in Ghana. At that time, I was thinking about breast cancer in Ghanaian women, because there is very little research that has been done to understand breast cancer in these women. And so we don't have answers, and we use treatment that is available, and they don't do very well. And during that time, I had the opportunity to meet some researchers from the University of Michigan who had come to Ghana to do some work. And from interacting with them, I understood that breast cancer that occurred in African American women was very similar to what occurred in the Ghanaian women as well. So this prompted my interest to for me to want to study more. And so I had the opportunity to come to the University of Michigan to do my PhD in cancer biology. And my PhD was focused on breast cancer in women with African ancestry. So for both continental African women and African American women and women with African ancestry everywhere. So wanted to understand the role of genetic ancestry in developing aggressive breast cancers, and if it's possible, how we can find better ways of screening, diagnosing, and treating breast cancer in women with African ancestry. And again, whatever we find in these women it has a potential to benefit women of all ancestry. It's not only going to benefit Black women, but it benefits everyone else. So for me, that was the reason why I wanted to study more about the breast cancers that occurs in Black.
So I know your focus is triple negative. Did this woman that was diagnosed while pregnant, did she have triple negative breast cancer?
That woman had triple negative breast cancer, yes, at that age.
Can you share what triple negative it is and why it is so challenging to treat?
When you take breast cancer, it is not one disease. So breast cancer is a group of diseases that affect the breast. And if you say cancer, it means that, or breast cancer, it means that the breast is growing at a faster, uncontrolled rate. And there are so many reasons that can lead to that. And there are some different markers, or there are different proteins or other markers that are expressed on the tumor that is used to diagnose the tumor, and it is used to treat it. For instance, if the tumor growth is controlled by hormones like the estrogen hormone, then we have estrogen receptor on the tumor that is so you refer to that tumor as being estrogen receptor positive. It means that the growth of that tumor is regulated by the estrogen hormone. The same with the progesterone receptor. And so when these you have the expression of these markers on the tumor, we call it the estrogen receptor positive or ER positive tumor, or the progesterone receptor positive or the PR positive tumor. Alternatively, you can have the HER2, which is a protein that is expressed on the surface of the tumor as well. And this can also be targeted to treat the tumor. But when you have a cancer or a tumor in the breast that doesn't express any of these markers, then we call it a triple negative breast cancer. And so by definition, this is a tumor that does not express any of the markers that we know and we use to treat the tumor. And so definitely it's going to be very difficult to treat it because it doesn't have what we have targets for. It doesn't have what we have treatment for. And so to treat these cancers, we have to target it with any available chemotherapy. So we have different combinations of chemotherapy that we throw at the tumor and hope that the tumor will respond. And so that makes it very challenging. And so by definition, triple negative breast cancer is diagnosing something that we do not know, not what we know, which means that it is possible that it is also by itself a combination of different diseases, which means that there is the possibility that there are other markers in these tumors that can also be identified and that can be used for treatment. And so it gives us room, or it means that there is a need to study this tumor as an entity in order to be able to find new ways of treating them, because now we don't know what we will be able to use to target it. And so the name itself means that it's the absence of what we know.
Interesting. That's a very good explanation. So what I find curious is that in the African American and the African population and in the Ashkenazi Jewish heritage population, there is a higher incidence of triple negative breast cancer. And I believe that's really what propels you forward in your research to understand why that is. Can you share a little bit of what you know now about why that type of cancer shows up in a higher percentage in those women? And also, have men ever been diagnosed with triple negative breast cancer?
So, when you take, thank you for that question. When you take triple negative breast cancer, it is among all breast cancer subtypes. It comprises about 10 to 15%. So if you take 10 women randomly, irrespective of their ethnicity, approximately 1 out of 10 women who have triple negative breast cancer. But when you break it down to ethnicities, white women have about 10 to 12% chance of getting triple negative breast cancer. African American women in the US have about 22 to 25% chance of getting triple negative breast cancer. In sub-Saharan Africa, where Ghana is, data shows that up to 80%, 80,
wow,
of the tumors are triple negative breast cancer.
Okay.
Among women who are genetically predisposed to developing breast cancer, that is women who have the BRCA mutation, those who have BRCA1 mutation, there is up to 80% chance that when they develop breast cancer, it will be triple negative breast cancer. And the BRCA mutation is most prevalent among the Ashkenazi Jewish population. And so that explains why the Ashkenazi Jewish population, women have a high percentage of triple negative breast cancer when they do develop breast cancer. The same, so when you look at it right now, from what we know, the women who are genetically predisposed to breast cancer, that is those who have the BRCA mutation, have an 80% chance of getting triple negative breast cancer. In the African women or women with sub-Saharan, Western sub-Saharan African ancestry, we don't know what predisposes them to begin with to triple negative breast cancer, but then they have up to 80% chance of getting triple negative breast cancer, which, I mean, by that begins to let us understand that there is a genetic predisposition, because the breast cancer that occurs in these women is very similar to those we know have genetic predisposition to getting breast cancer. That is the first one. Secondly, when you take factors that are protective for cancers, for breast cancer as a whole, like if a woman breastfeeds, has a lot of children, breastfeeds the children, has early menarche, has give birth to the first child early, these are all protective factors, but then they protect you from estrogen positive breast cancers, but they don't protect you from triple negative breast cancers. When you, among African women, all of these protective factors are present. They have many children. They start having children at an early age, but then so it doesn't protect them from triple negative breast cancer, which means that the factors that are predisposing these women to getting breast cancers is different from those protective factors that we know. In a study that we published last year, we realized that indeed there is we found a genetic marker that predisposes women who share ancestry with Western sub-Saharan African women, so it predisposes them to getting triple negative breast cancer. So there's a genetic predisposition to that. And so, and if you take African American women here in the US, most of them came to the US through the transatlantic slave trade. And that went mainly through Western Africa, through Ghana, where we have all the slave castles and all the structures that were used for the slave trade. And so there's a shared ancestry between African American women in the US and African women, or Western Sub-Saharan African women, which means that whatever genetic markers that we are seeing in the African women is shared with African American women. And so there is this genetic predisposition to developing triple negative breast cancer. And the protective factors that protect women from developing breast cancer doesn't protect the African woman as much as it protects other ethnicities as well.
So I know that when I was diagnosed, I had my first child over 30. I mean, now it's over 30 is considered a factor in getting breast cancer.
Yeah.
Being overweight, a high BMI, a poor diet. But it's so interesting that what you're saying is that if a woman of African American ancestry, or maybe even Ashkenazi Jew, could have their first child before 30, nurse their children, have a great BMI, exercise, have a healthy diet, but because of the genetic predisposition, they could be diagnosed with triple negative.
Yes. So all those factors you mentioned protect you from estrogen positive breast cancer, but not triple negative breast cancer. And so there is the hypothesis that it might even, the factors that protect you from developing the estrogen receptor positive breast cancer might actually be increasing your chances of getting triple negative breast cancers.
Wow.
Yeah.
So your focus is on this triple negative breast cancer, and you have a very specific plan to carry out your research. Can you share what that is and how that's going to work?
Because triple negative breast cancer is most prevalent among Western Sub-Saharan African women and Ashkenazi Jewish women, we believe that even though this disease affects women of all ethnicities, and it has been very difficult over the years to find a solution or to find new ways of treating triple negative breast cancer. The best way for us is to study the disease among the population with the highest prevalence. And once we find a solution among that population, it will be very easy to be able to translate it to other ethnicities as well. And so the the research group, the program that we have here at Henry Ford called the Precision Medicine for Aggressive Breast Cancers, is an institution that is focused on finding new ways of screening, diagnosing, and treating triple negative breast cancer. And we are doing that by studying triple negative breast cancer in African American women here in the US in comparison to the tumors in African women, and studying the Jewish women here in the US in comparison to women in Israel. So the model we are using, for example, if I'm using the African American model as an example, if you take an African American woman, there is a chance that she wouldn't be completely African. There will be other ethnicities in her ancestry. So an African American woman who is self-reported African American woman might be maybe 50% West African, might be 30% European or other ancestry. So, and there's a very wide range of the percentage of African ancestry in the genome. But so it is very difficult to tell by taking the African American tumor what the role of the African ancestry is in controlling how the tumor or her breast cancer behaves. But that can be understood better if you took the tumor from a pure African woman who doesn't have any other ancestries admixed in her genome. Then you study that tumor and compare it to the African American tumor, for example. So if you take an African American sample and the woman, you check her ancestry, you are able to tell that the 50% is from West Africa, and another one, the 50% is from East Africa. And you have pure East African tumors, pure West African tumors. We are able to compare that and be able to determine that because she's 50% from West Africa, these are the drugs that are good for targeting breast cancers from the women who are with pure West African ancestry. I mean, there's a higher chance that she will respond to those drugs when she's treated based on her ancestry than if you treat her with the drugs that are better for people who share ancestry with East African women, for instance. So we are trying to do study these tumors and find drug or treatment based on their ancestry and based on the genetic mutations or markers that are regulating the tumor. And so by studying the women who have no admixture, who have no other ethnicities mixed in their ancestry, we are able to determine the precision, and it's called precision medicine. We are able to find the best way to treat women who share ancestry with those people. And again, this is not only for African American women. Considering that the African continent is the oldest continent, it's believed that every woman shares some ancestry with or has some African ancestry. And so if we are going with the ancestral route, then whatever we are finding, we believe will benefit women no matter what their ancestry is.
So I know that all research like this is a long time. It takes a long time. Can you share from the beginning of when you decided to start this research to your projected end date where you have some data that you can share, why it takes so long and why it's so expensive?
Thank you. So I can talk about where I began. I don't know where it will end, and we are beginning to find new biomarkers for screening and for treating. I started this work at with Dr. Wisha Maswisha and Dr. Sophia Maraiva at the University of Michigan as part of my PhD. And for my work, I had to, because I'm a surgeon and I worked in Ghana. I traveled to Ghana. I operate on the women with consent, and I brought back the tumors. Some of the tumors on dry eyes flew in, usually within 24 hours.
Wow.
Bring the tumors here to the US, and then implant these tumors in the lab so that they will grow for us to be able to study them. And so even that process alone, to be able to travel in and bring back the tumors, each trip costs tens of thousands of dollars to be able to bring these tumors to study. But the reason why we had to bring them here was, one, there isn't infrastructure there to be able to do the complex experiments that we need to do to find these, to the solutions that we are finding. And we needed to make to do a comparative analysis. So when you take research, one, there are so many, there are components that go into it. One is the personnel, the people who have to do the work. These people have to be paid. Mm-hmm. So if, for instance, you come to the lab right now, we have partners, and international partners and local partners. We have people here at the Henry Ford Institute who have to go to the clinic, talk to the patient, consent to the patient, and those who consent to be part of the study will take their saliva, their blood, their tumors. So there are about 2 or 3 people who are paid full time all year round, and that is their work. And then there are people in the lab who have to receive these tumors, process them, and do experiments on them. There are people who are in maybe the sequencing core who have to do the genetic analysis on the tumor. So even in that just one lab, we have about 10 people who have to be paid full time to do the work. Then you need equipment to do this work. And there are so many different equipment that are needed. There are the microscopes that you need, the sequencing equipment that you need, the incubators, and the prices of these equipment range anywhere from maybe $20,000 to $500,000 for one equipment, or even more, to be able to do. Sometimes there are core facilities, like in the University of Michigan, you have either the institution purchasing the equipment to be shared, or other researchers coming together to buy so that they can share, so that it reduces the cost of doing their work. And then if you find something and it has to move to clinical trials, or you have to develop components or drug for it, there are so many other industrial components that come into it. And all of this is money, and it takes a lot of time, sometimes 5 to 10 years before you identify a drug and move it through clinical trial for you to be able to use this for patients.
So I want to be really clear about consent, because here in the US, so many people of color have been used in scientific research without consent. So the people that allow you to sequence their tumor, they've signed some kind of document, I assume.
There's a lot that goes into that. It starts at a national level. There are regulations that you have to comply to. There are a lot of training processes and programs that you have to go through as a researcher, and all the people that are working, you have to go through those training. And then there is the institutional review board. Mm-hmm. You have to present your protocol to. This is a panel that will go through and make sure that nothing that you are doing is going to breach the patient's personal information, or is going to go against the patient, or going to be harmful to the patient in any way. And once that is done, and that process is approved, then it comes to training the people who actually speak to the patient and explain the process to the patient. And they have to make sure that the patient is not being forced to be part of it. It has to be done out of her free will. And when that happens, it will have to make the patient understand that whether or not she partakes in the study doesn't affect her treatment in any way, and she's free to get out of the study at any time. If at any point in time she decides that she doesn't want to be part of the study, she's not obliged to be, and it doesn't affect her treatment in any way. So you just explain what the benefits are, and that we are trying to find new treatment that might not even benefit her at that time, but might benefit other people that she might know. And so it is very important. And as a Black woman, I know, and I really understand the importance of making people comfortable and understanding fully what goes into this. And it goes beyond this, even clinical trials. It's very difficult to get women of color to partake in clinical trials because of the historic experiences that we've all seen, and the way women have been experimented in the past. So, but we are hoping that in partnership with community leaders and people that the community trusts, will begin to build trust in the community for inclusive research. Because if the research we are doing does not include all races and everyone is not represented, then the solutions we find are not going to benefit all women equally. It will only benefit the women whose tumors are studied and who are experimented on, if that makes sense. So if they are not represented, then the cycle goes on, and whatever treatment comes out doesn't benefit them equally as well.
So I know you talked about your sample size for both the African population would be about 500 tumor samples and 500 in the Ashkenazi Jewish population. Can you explain why that number is kind of a sweet spot?
Yes.
So why not 1,000 and why not 100? Something like that.
Yes. So in order to begin to find possible new treatment, we need to study the tumors and the normal sample from these populations. The larger the sample size, the higher your chances of finding something that is going to be beneficial. But then there is the issue of cost. So now to sequence one tumor costs around $500. Then it comes to the analysis that goes with it and everything. So in order for you to fully understand one sample, it is going to cost around $1,000. And so we have to look at the capacity we have, but at the same time, what we have has to make significance. And so we are looking to sequence an initial 500 sample from each ethnicity. And we are looking at this because we are that is a number that we hope to be able to find the funding to do. And it is also a large enough sample size that whatever we find will be significant enough for us to be able to extrapolate it to the larger population. And with that, we'll be able to optimize our experiment and be able to move to a larger sample size. But to begin, when you are beginning, again, because of all the other factors that comes into play, you want to be able to peg your number at a point where you have the significance, but it's not too expensive that you cannot do it.
Well, I love that. That's a great answer and explained very well. So let's talk about how you fund these studies a little bit. I know that, do you get funding from, say, the American Cancer Society or the National Cancer Institute or National Institutes of Health or the Department of Defense? Are you looking for money from private individuals? I know that you mentioned that you're also collaborating with other health systems. So explain how a person like yourself who's running a lab and has this program, how do you go about funding the millions of dollars you're going to need just to get yourself to the place where you might have something you could bring to a clinical trial?
Henry Ford, thank you very much. Henry Ford as an institution is very interested right now in finding a solution for triple negative breast cancer in women of African ancestry, because we serve a very large African American population. And so the work I am doing right now is solely funded by Henry Ford Health Cancer Institute. So the leadership felt the need that this is time, it is time for us to address all the inequality in the system and focus on the tumors in the women with African ancestry and triple negative breast cancer, which is very aggressive. And so they have invested initial $1 million+ in this work, but that is just the seed money to begin with. I partner with my development team. We have also begun to look for donor funds to continue with their work. People who understand their work in breast cancer and have the interest to find solutions for breast cancer, irrespective of their ethnicity. There are people like that who are beginning to contribute to the work. In addition, we are looking for funding, government funding as well. So we write grants, and usually you get to that point when you begin to get data. So once you get data, then you are able to apply to donor, to government fund and also foundation funds as well. But currently, the work is funded almost 90% or more by Henry Ford Health System or Henry Ford Cancer Institute, because the institution sees the need to begin to do this work.
So we have just a couple minutes left.
Yes,
you have given us, imparted so much information. So I just kind of want to review what really motivated you was treating this young mother who was pregnant and had triple negative breast cancer, and when she heard her diagnosis, collapsed and knew it was going to be her death, that she would die. And so then you became really passionate about trying to understand this. And you came to the United States where you felt you could get more support for the work and more training. You also said that you were a surgeon. Do you still perform surgery?
I do still perform surgery, yes. When I'm back home in Ghana. Here I don't. Here I'm focused mainly on research, but I still have my license to practice. But right now I just do research, breast cancer research. So I'm MD PhD.
So let's pretend that there's a number of women in front of us who have triple negative breast cancer. What would you say to encourage them at this point?
If a woman comes to me right now with triple negative breast cancer, I would tell her that there's currently we have studied the tumor as much as possible. There are drugs that are available that are used to treat that is available that we can use to treat her tumor, and we are very optimistic, and that we are going to do everything that is available for us to give her the optimum care. That as a woman, she should have faith, and she should have be ready to be bold and fight the disease, and that she has it in herself that this disease is not going to overcome her, and that there are so many women, there are so many people who are ready to support her in her journey to fight the disease. And when we know, not if, but when she overcomes it, she will join us in the fight for a better treatment option for women who have this disease. That we believe that we don't have all the answers yet, but we have the best possible treatment available right now. And there are new options that are coming up every day, that we are going to give her the best possible treatment that is available. And that the disease is not going to overcome her, but then she has it in her to fight it and join the thousands of women who are cheering her on her journey.
So Dr. Jiagge, thank you for your time. If somebody wants to reach out to you, are you able to provide an email or some way to contact, or would you like them to come through the Pink Fund?
I think they can come through the Pink Fund. You have my email, and if you I mean, using it, I mean, you are free to give my email out as you deem appropriate.
Yes. So if anybody has any further questions for Dr. Jiagge, whether you're on live with us now or you see this at a later time, please feel free to send an email to info@thepinkfund.org. That's .org. And we'll be sure to get your question to Dr. Jiagge and return with an answer. And Dr. Jiagge, thank you so much.
Thank you.
Keep track of how you're going and have you back again. Have a great Thursday evening, everybody, and we'll see you next Monday. Again, these segments on Facebook Lives are brought to you by Exact Sciences, the makers of the Oncotype DX test. Good evening.